Okay, again, thank you very much, everyone. Today I'm going to talk about our recent study that deals with the genetic diversity and endosymbiont prevalence of Kelly citrus thrips in order to shed some light on its source of invasions from Australia to its invasive regions. But before I start, I would like to acknowledge the traditional custodians of the lands which I have conducted my experiment in. And today, this is the land of the Dora people, and I respect the elders, past, present, and emerging. Alrighty, so Kelly citrus thrips, or scientifically named as Pezzothrips kellianus, is an important pest of citrus in Australia that can severely damage flowers and fruits of different citrus species. And adults and larvae of this species, as you can see in the slides, it's generally sheltered within the citrus flowers and feed on pollen and very young fruits. And their feeding activity and on the surface of the young fruits can cause patches of scar tissue and necrotic area on the apex of fruits, as you can see on the picture in the center. And this damage can lead to the downgrading of the quality of the affected fruits and reduce the marketing value. And another important aspect is that this species is also abundant on Australian native frangipani during early spring, which is suggested to be one of the main host species of this pest. Pezzothrips was first described from Queensland, but it has also since been found in other parts of the world, including New Zealand and Mediterranean region. And the great work by Lawrence Monn's team suggested that this species is native to Australia. However, it was not genetically confirmed until very recently that Zung Nguyen in our team has confirmed the Australian origin of this pest species. And it was actually, this was inferred by higher mitochondrial and nuclear diversity that has been found in Australian population compared to populations from New Zealand and Mediterranean region. And the previous study in our team has shown that the population of Australia are infected with the maternally inherited bacteria Wolbachia cardinium. And in contrast, the population of invasive range in New Zealand and Mediterranean region are only cardinium infected. And it is worth mentioning that both of these endosymbionts can independently cause cytoplasmic incompatibility in this species. And previous work by our team revealed independent colonizations events of New Zealand and Mediterranean region as population of both invasive regions harbor diverged mitochondrial haplotypes without any other haplotype diversity within these regions. And however, this work only analyzed a few populations in native and invasive range with limited number of thrips species that thrips specimens that collected during that time. And actually, this did not provide a full picture about the genetic diversity and endosymbiont prevalence in invasive ranges. Therefore, we believe that more genetic analyses are needed to unveil the source population within Australia for both invasive and native ranges. And in order to identify the source population within native Australian range, we address the genetic diversity using CO1 mitochondrial markers, along with the analysis of infection prevalence of Wolbachia and Cardinium in its Australian range. And also, and this was based on extensive sampling that we have conducted from different populations in Australia and New Zealand and Mediterranean region. And as you can see in the map, first of all, I have to mention that the purpose, in order to meet our purpose, we collected the thrips individuals from more than 35 populations in 11 designated regions, as you can see in the map in Australia. And thrips specimens were subjected to DNA extraction, and they were direct sequenced for their CO1 genes, and each population also were screened for Cardinium and Wolbachia infection afterward. And in this map, you can see that the infection prevalence of Wolbachia and Cardinium in Australia, which in the red color denotes double infection, blue color is Cardinium only infection, and yellow one is Wolbachia only, and finally the white one is infection-free status. And we showed nearly 100% prevalence of Wolbachia, 100% prevalence of Cardinium in all population that we assessed. However, Wolbachia prevalence showed a very different pattern with New South Wales population and Queensland one showing overall the highest prevalence of Wolbachia, and even Wolbachia was higher in Eastern Coast region compared to inland population. with more than 90% overall prevalence in mid-north and central coast and Hunter Valley and northern rivers. And analysis of more than 60 tested individuals of Victorian population and Western Australian populations showed complete absence of Wolbachia. And the Wolbachia was also shown in very low prevalence in Southern Australian, with some populations even showing complete absence of Wolbachia, which is very interesting. We further did the symbiotic screening and CO1 analysis for 5 populations from Northern Island of New Zealand and from populations, 5 populations from Mediterranean region. And our results showed that all populations from New Zealand and Mediterranean region were fixed for cardinium, but Wolbachia was not detected at all in any of our PCR assays. And overall, we characterized 12 haplotypes from Australian populations. And according to our phylogenetic analysis of Pezethrips using maximum likelihood, we show that these haplotypes are monophyletic when compared to the other related species. And we showed the highest haplotype diversity in Australia, and they were particularly high in Victoria and South Australia, where Wolbachia was absent or was low in prevalence. And moreover, our extensive sampling in New Zealand and Mediterranean region still showed no CO1 diversity, which suggests that the population there established after a single haplotype in each region, the establishment of each single haplotype in each region. And however, the extensive sampling from Australia as revealed previously found haplotype 1 in Mediterranean region and haplotype 5 in New Zealand, and we also found the same haplotypes in Victoria as well. And according to our haplotype network analysis, we have shown that the lowest haplotype diversity for Queensland and New South Wales population. We also showed haplotype 7, as you can see on the right-hand side. This was the most frequent haplotype in Australia, which is dominating across New South Wales. And this haplotype showed more than 95% infection with Wolbachia. And haplotype 7 and haplotype 8 clustered closely and are found in New South Wales and Queensland. However, haplotype 2, 4, and 8, they showed less than 60% Wolbachia infection. Whereas we detected the highest haplotype diversity in South Australia and Victorian population with haplotype 5 and haplotype 1 and haplotype 5A and haplotype 1 and 1A and 1B, clustering into the closer network with all haplotypes lacking Wolbachia. And haplotype 2 and 2A mainly occurred in South Australia with haplotype 2A lacking Wolbachia. And interestingly, Victorian population shared haplotype 5 and with New Zealand population, and haplotype 1 with Mediterranean region, suggesting that Victoria is likely the region where Pezethrips has invaded from in New Zealand and Mediterranean region. And Western Australia may possibly also have been established by individuals from Victoria that has actually shared the same haplotype with Victoria, which is haplotype 5. And our study only showed one strain of Wolbachia and cardinium in all tested populations. But one interesting finding, as I mentioned, is that the New South Wales populations predominantly infected by haplotype are occurring by haplotype 7, which is infected more than 95% Wolbachia infection prevalence. However, higher mitochondrial genetic diversity of South Australian is linked with the absence or lower frequency of Wolbachia infection in these regions. And this suggests that this single strain of Wolbachia may be driving a selective sweep of mitochondrial haplotype in Pezethrips. And therefore, Wolbachia might have been acquired in native range of this species, and probably more recently and more recent than cardinium, probably via horizontal transmission, and that resulted in a very low haplotype diversity, as you can see in the Eastern Australia. And finally, our study showed a shared haplotype in Victorian population and New Zealand population and Mediterranean region. And this primarily suggests that Victoria as the source invasive candidates. However, we believe that an alternative location of the source population could be other region of Eastern Australia which have since experienced an invasion of Wolbachia and then thereby linked replacement of haplotype after Wolbachia establishment. And therefore, we believe that population genetic analysis across Australian population and in investigation of the geographic population structure may provide a more concrete answer regarding the source population of this species in Australia. And currently, we are analyzing population genomics data sets to compare the genetic diversity and geographic population structure in Australia and New Zealand on the same samples that we have collected and have done the haplotype analysis. And for this, we are using diversity array technology or DART sequencing to further confirm the Australian source of invasion of pezzothrips in New Zealand and Mediterranean region. And finally, I would like to acknowledge our team in the Hawkesbury Institute for Environment and all the people who have helped me in this project by sampling and other sources, and the funding organizations that supported this project. Thank you.
Wonderful. Thanks very much, Alihan. Does anybody have a question for Alihan?
I've just put a couple in the chat.
Oh, right. Thanks, Perrin. Yeah. Would you like me to read
wondering,
it out? Like ask? Oh, sorry. There you go. Go for it.
Oh, yeah. I guess I can just read it out. Yeah, I was wondering what Wolbachia strain this one was most closely related to. Do you think it? And if you think it came via horizontal transmission, do you think it? You know, is it an insect that's closely related or?
Yeah. We believe that it has been transferred via horizontal transmission because it's highly closely related to the Wolbachia that is a strain that from the supergroup B, and it's fairly close to another species. But this is a complete new strain that has been previously found by Zung's project in our previous study, but it's fairly closely related to the Wolbachia in. If I'm not wrong, it's fairly close to the Wolbachia in Tetranicus artica or spider mite.
Right. Okay. Yeah. And my other question was, you know, why do you think you're finding this difference in Wolbachia infection frequency in these different locations, especially in New South Wales? You've got the very big difference between coastal and inland New South Wales.
The thing is that there's a haplotype difference in the inland and the haplotype in the coastal regions. Well, it has not been shown in the map, but the haplotype in the eastern part and in the coastline is actually all more than 95% haplotype 7, whereas the more we get towards the inland area, the haplotypes change into haplotype 2 and less haplotype 7. That's why I assume that Wolbachia has been so far established more in haplotype 7. And one of the reasons it could be also a temperature impact, and probably the other haplotypes in this region are infected with Wolbachia, but temperature impact might have resulted in the loss of Wolbachia as well. Mainly because we conducted the experiment in that area in the summer, which was fairly hot.
Interesting. Thank you,
cool.
Alihan.